To get you an artificial pancreas system that you don't need to bolus for meals, we need to have two main things. One is sensors, more accurate and more reliable sensors. And also, too, we need faster insulin. For an artificial pancreas system to work, it has to respond to the food that you eat by measuring, sensing the increase in blood glucose, and then to deliver insulin as a result of that. And as you know now, you have to bolus, 15, 20 minutes before a meal. So basically, we need faster insulins. So to get us to a point where we have highly accurate, highly reliable continuous glucose monitors, JDRF launched, in partnership with Helmsley, launched the Glucose Sensor Initiative. This was a substantial effort to accelerate the delivery of more accurate and reliable glucose sensors to market. And we've made the first grants. First one, again, in a press release, and this is dated June 1st, was a partnership between JDRF and Medtronic to develop the next generation CGM device. And this now is, right now we're focused, the CGM devices all rely on glucose oxidase. We're also looking at, can we have other chemistries and so on to have kind of redundant sensing using two different chemical approaches. So we have a partnership with Medtronic now. And also, too, we have a partnership with BD. We announced in early June as well. So we feel very confident that with these partnerships, that we're actually going to be able to deliver in the years to come very accurate, reliable CGMs for closed-loop control. That's second-generation sensors, second-generation artificial pancreases. How about faster insulin? So we got half. We think we can solve the sensor issue. Now, how about faster insulin? Well, there are different ways that you can do this. You can modify the insulin, where you could, insulin is hexameric. You can actually deliver it as a monomeric insulin. It's a lot faster, so you inject it. That's a strategy that people are looking at. But there's also, you can actually accelerate insulin by the way you deliver it. And some examples of this that we're looking at in terms of accelerating insulins for the second generation and the third generation is a simple one, actually, and that's the InsuPatch. A simple concept that you can actually heat around the infusion site. A small heating element right where you infuse increases the blood perfusion in that immediate vicinity, and that actually, if you can see here, you can see this without InsuPatch. See how kind of flat and long. So delay of insulin appearance in the plasma, slower clearance. Now look at the InsuPatch. Much faster appearance and much faster removal. So these are strategies for acceleration, and this is actually a lot of work being done now at Yale, which we're funding. They're looking at the various parameters, the delivery and so on, the heating parameters, but also, too, they're looking at this in the context of closed-loop control in their algorithms. Another strategy, probably it's not everybody would want to do this, but this is a very, very fast approach, and this is the diaport approach. Now, what this entails is a port that actually delivers insulin directly into the intraperitoneal space. This is extremely rapid, but it requires surgical implantation of this port device. But look at the speeds. The open circles correspond to the appearance in the plasma of insulin if you gave a subcutaneous bolus, say through a pump. The closed circles, here the full circles, correspond to the diaport. Again, much faster, much more rapid delivery. So these are strategies. This might not work for everybody. There may be a subset of people that may want to have a diaport approach, but this will actually provide much more rapid insulin action and delivery. Another approach is it's actually part of a partnership we have with BD, and this is microneedles. What they found is that If you have a very small needle, and you can see here the size of the needle here, a very small needle as opposed to a long needle, it goes into the intradermal space as opposed to the subcutaneous space. There's apparently more perfusion there, a more rapid delivery of insulin to the body. So we're actually exploring this as well with BD in a partnership, looking at acceleration with microneedles and how that could be used in a closed-loop setting. So again, multiple strategies to see if we can accelerate insulin delivery and insulin action. And then here we have an example of another out-of-the-box example, and this is inhaled insulin. So you know when you look at food, even before food touches your mouth, your pancreas is releasing insulin. This is how fast it works. Inhaled insulin actually approaches that speed. You can actually have very rapid delivery of insulin with inhaled insulin. So we're looking at that too in the context of how that could work in closed-loop control. Not shown here are actually other strategies. For example, there's a new idea of enzyme called halozyme, which you can co-infuse with insulin, and that actually breaks down the extracellular matrix to actually more rapidly cause permeation by insulin. And that's something that's another strategy as well. Okay, third generation systems. So here we talked about first generation, second, and third. Well, third generation systems, the challenges here are, we talked about glucagon and the need for glucagon. Well, there's no commercially available soluble pumpable glucagon. So JDRF is actually leading the efforts there to actually develop this. Also, if we're going to deliver both glucagon and insulin, we have to have obviously a dual-chambered pump. Okay. Well, to address the glucagon issue, we launched an initiative, and that's underway right now, where we're funding various grants to identify soluble versions. Everybody knows about glucagon, a rescue pen, for example. But we need something that's almost like a soluble, like insulin, that can remain in the pump and not fibrillate and be compatible with the pump. Well, then here's another press release. We actually, there was a paper by one of our consortium members, and they've actually been looking at glucagon, and they actually were working, and they've actually believe they have found a very good soluble, pumpable glucagon. Still a lot of work to do, but it's very exciting that they've reached this stage already. Just want to touch briefly on glucose-responsive insulin. So the idea behind glucose-responsive insulin is that you'd have an insulin that would be completely inactive. You'd inject it into the body, and it would only become active when your glucose levels raised above a certain level. And that increase in glucose levels would actually cause the insulin to become active and then bring that glucose level back down below the level of the insulin being active. That's the concept behind glucose-responsive insulin. So we funded Smart Cells a number of years ago, and actually the NIH as well, and we de-risked that company. It was a very small company with a very, very cool technology, and they've been bought by Merck, and we're very excited about that, and Merck has now has committed to take that forward. So we're looking to see, we're hoping and anticipating some very good results from there. But also at the same time, what we've done too is to have multiple shots on goal. We've also launched a glucose-responsive insulin prize. This is kind of new for JDRF because, we used to actually just go to the academics, to have academics kind of do the research. But right now we go to the academics, we go to industry, but we've also done crowdsourcing now. Opening the question now to the broader community. To see who has ideas, who has suggestions for glucose-responsive insulin. We did this, and we had a tremendous response. Lots of really, really good ideas. 50 applications received. We reviewed them. All the submissions we had on the review panels, we had biochemical engineers, polymer scientists, and such. And we're going to announce some very cool results, some very cool ideas very soon. So more to come. So in summary, I'd just like to say that there's a tremendous amount of momentum in the artificial pancreas. I've been working in this now for about four and a half years, I think it is, and I've never been more excited. the FDA, the outpatient studies, the new CGM. I mean, even before this sensor initiative, all the nice new sensors that are coming out on the market. And also, too, the momentum. We just came back from the ADA meeting, where JDRF held a workshop in conjunction with NIH. We had a tremendous amount of interest. The FDA were there. The FDA have changed completely. They're so interactive now. We have a tremendous amount of interest from companies and also academia. So, thank you very much.
Thank you, Dr. Pragnell. Dr. Pragnell is going to take questions now. We'll do that for maybe 5 or 10 minutes, and then we'll switch over to our other research presentation in South Dakota. And at the end of that, we'll take more questions. And if something occurs to you that you still want to ask Dr. Pragnell, he'll still be here. So I'm going to start with. Can I start with South Dakota to see if they have any questions there first? Or did that just throw you off? Look at all our friends there. Nothing there, Katie? Nothing.
Nope, I think we're okay.
Okay, do we have people out in the conservatory? We do. Oh, I see them. We do. You guys got to sit in here, so you'll have to go last. Nope? Okay, so any questions in the room? Oh, see. I'll give you a mic so that we need the mic so that the people out there in South Dakota can hear you. Talk loud.
Sure. It's a little curious to me that JDRF would decide to partner with Medtronic and give them money for studies when they seem like such a big company with deep pockets on their own. Does anyone have any perspective on that?
I do, yeah. So they do have deep pockets, but not in the path that we want them to go in. The whole focus of JDRF's efforts is to drive the research towards artificial pancreas, and it was a real gap. We want the companies to develop CGMs for an artificial pancreas. We want to set the priority, and the way we set the priority is through partnerships.
I recently got to spend some time with Jeffrey Brewer, our CEO, and the way he explained it to me was when we make these grants to our industry partners or work with industry partners, we're buying a result. And in the same way that we used to buy results from academia when it was basic research, we're buying results in a different way from industry partners. They only get paid if we get the results that we're looking for. So it is important to understand that. And sometimes an industry partner is in a better position to provide those results than maybe an academic partner might be. So that's, if that helps to kind of explain how that happens with a company like Medtronic. And then in some other instances, it's really providing some seed money to a small startup company that wouldn't be able to take this great idea they have and push it forward if they didn't have some extra capital to do that. And in a lot of those cases, when the result is accomplished, JDRF will get its money back. So that's another important thing to understand.
Yeah, I mean, the important thing is that, the way JDRF funds grants, it's all milestone-based, heavily monitored. We don't just throw money over the wall. With all of these partnerships, they're carefully monitored. Money is only released when they deliver on their milestones.
Go ahead.
Hi, I used to work for Desatronic 13, 14 years ago, and was excited to hear about the update on the Roche program, the enteral perinatal. A year or two ago, I asked the Reach and Teach if there were any updates on that, as I've been wearing an insulin pump since the mid-1990s, and after so many years wearing an insulin pump, experienced site issues and absorption. You mentioned on that project it may only work for a small subset of people. I'm just wondering if you could share a little bit more about that, or who it might be a good candidate for.
Yeah. So it's the diaport, and this is the intraperitoneal delivery of insulin. And really, it's a port. It's like almost like if you think of somebody, for example, who's on dialysis, has a port where they go for dialysis, the same kind of thing. It's originally intended for people with brittle diabetes, very hard to control. When they go on the diaport, they get spectacular control because they get almost instant delivery of insulin, no hypoglycemia, and so. But the fact that it is a port and it's surgically implanted, there are people that they're not going to want to have that, and so it's only for people that obviously want to have that, and those who are brittle. I think ultimately, though, we may find broader adoption. We are actually funding research right now in France, Dr. Eric Renard's group, and they're actually doing closed-loop studies, artificial pancreas studies with the diaport. And so if we see great results with that, will be a strong incentive to actually go and actually get the diaport.
Go ahead.
Okay, my question is, I've been actually a diabetic for 49 years. Next year will be my 50-year anniversary, so I'd be a good guinea pig if you ever need one. But what I'd like to ask is, and this is what puzzles me, as you were talking about the different newer systems that are going to be coming up, and just about how, because I've had numerous hypoglycemia instances because I have what you call the unawareness and that, but what really puzzled me with all these pump companies that they don't have, like I've talked about this, and I said, if you've got the technology to do what you have now to make these pumps and do what they do, why they haven't been able to create a pump where you can do like your phone, raise the volume or lower the volume. Because I understand why they don't want it too loud when you're in a business meeting or something like that. But I'm just wondering why they never developed something like, because that to me would be a pretty inexpensive and a simple fix, I would think. But
with alarms? You're referring to alarms? Yeah, actually, recent data coming out of Tim Jones' group in Australia said that a lot of people don't respond to alarms. This, if you have the same alarm, same sound, some of the recommendations are maybe change up the alarms. Different alarm or something like that, something to catch your attention. But really, we want to get beyond alarms. We want to get to a point where you don't need to worry about alarms. That the glucose, the sensor will tell the pump to deliver more or to stop delivering. So, yeah. But you're a good point. I think the key there is to vary the intensity and the type of alarm so that people kind of are more aware of it.
Yes. I realize that what you're talking about isn't a cure. It's not the cure stage. It's the treat stage. And you probably can't answer this specifically. Our son developed type 1 when he was 20 months old in 1974. And a doctor at NIH had told my mother, there'll be a cure for diabetes within 5 years. So we've all heard that. That was in 1974. So do you have any remote idea of when this closed-loop system might actually be available commercially?
So the first stage is available right now, the low glucose suspend. The predictive suspension, predictive hypo, outpatient studies are underway. Again, it's more regulatory than science. The hyper minimizer, we're very confident that's going to be available, and that's why we went into a partnership with Animas. Again, it comes down to regulatory. That's the real issue. The science is actually looking very good. I mean, I see a lot of the data and so on. It looks great. The whole thing is that you cannot make predictions unless it's regulatory. We were held back. We funded grants in 2009 for outpatient studies, and we're just starting them now. And it was not because of the science; it was all regulatory. So it's we're kind of cautious about making predictions, but we know that those issues are at least for the stage 1 through 3, it's regulatory. Stage 4, 5, and 6, we need technology. So those are probably push a bit further out. But with the pace of research now and the momentum and the excitement, with that level of momentum, and and this is all JDRF driven, all the excitement, things move faster, a lot faster. But in terms of an actual date you will receive it, I know we'll receive the low glucose suspend quite soon. For stage 1, 2, and 3, I would say within the next 5 years.
Hi, I had a question about the artificial. And I noticed there were no centers in the Minneapolis, Dakotas area. Will there be any opportunity for residents in this area to participate in the outpatient trials?
Yeah, so the trials right now are small pilot studies. They're centered around the academic institution that's running them. So, for example, Boris Kovachev in University of Virginia is doing a lot of the controlled terrain studies. So he has just selected a small number. So it's Virginia, Sansom in California, Montpellier in France, Italy, Padua in Italy. There's no barrier to participating in the trials, but the challenge here then obviously is the inpatient studies have been very labor intensive so far. We had to have the inpatient studies required like a doctor to be there on site the whole time, and they were very, very expensive, and they were only able to do it at very, very few sites. The outpatient trials are now morphing to something very different, where, for example, as I mentioned, Howard Zisser in Sansom is doing 5 patients this week all at once, and they'll be doing more and more and more. That will open up probably more centers and so on, more opportunities at different locations. But right now it's centered at those locations. But to enroll in trials, though, you really go to our website and you can actually see what's going on and seeing, and not just in artificial pancreas efforts. I mean, really get involved in other trials as well related to type 1 diabetes.
Okay, I'm going to check one more time with South Dakota to see if there's any questions there, and then we'll move on. And Dr. Pragnell will still be here, so we can continue to ask questions about the treat area. You good, Katie? You good, Katie?
Anybody have any questions? Got a microphone.
Okay.
Okay.
Thank you very much.
Great. We're really excited to be joined next by Dr. Alex Rabinovitch. He's joining us live tonight from the Sanford Research Center in Sioux Falls, South Dakota. Dr. Rabinovitch is the senior scientist of the Sanford Research Project and a professor in the Department of Pediatrics at the University of South Dakota. His most recent research has led to the discovery of a treatment that can regenerate insulin-producing pancreatic islet cells and cure type 1 diabetes in mice. He is now the principal investigator of this regeneration therapy being tested in a clinical trial in human patients with recently diagnosed type 1 diabetes. Please join me in welcoming Dr. Alex Rabinovitch.
Well, thank you. It's getting a little late, and I certainly sympathize with you. I know you're very motivated, but I'll try to be as brief as possible. Perhaps 20 minutes to get through these slides. Now, I want to do 2 things. First of all, tell you a little about the background, the basic science that we did that led to the clinical trial, and then the aspects of the clinical trial itself. It all started, as far as Sanford is concerned, as you see here, in June 2008. What happened then was that T. Denny Sanford wanted to put some money on the table to, in his words, cure preferably a pediatric disease. And JDRF made, I think, the best pitch as to where the money should go. And that's why we're at Sanford particularly indebted to JDRF for their input as to how important it was to direct the funds that he put on the table to work towards a cure for type 1 diabetes. Point one. Point two is that they went even further. And among the portfolio of ways that JDRF thought that diabetes might be cured was beta cell regeneration. So you see what it says there, that the focus would be beta cell regeneration. Now, what do we mean by beta cell regeneration? Beta cell regeneration, you see there on the bottom, is simply one of the ways to replace the beta cells that produce insulin that have been destroyed by an autoimmune process. You've heard from our Minnesota speaker about what you see, number 3 there, the artificial pancreas. Now, the artificial pancreas can replace the insulin-producing beta cells. If the artificial pancreas treatment, it's been called a treatment, were perfect, it would be a cure. Other ways that you might cure by replacing the beta cells would be by transplanting them. They might be human beta cells or they might be porcine beta cells. And those are other areas that are being worked on. So let's talk about beta cell regeneration. That's replacing them from within the body. So the first question to ask is this all a fantasy, that you can get the lost beta cells within the pancreas to regenerate? The answer is no, it is possible. It is possible because it does happen. It even happens in adults. Not adults that have diabetes, but adults that are either overweight, obesity, during pregnancy, and even after gastric bypass surgery. So there are 3 natural situations, physiological if you will, where we know that even in the adult human, beta cells can regenerate or grow. Let me show you an example. This is the pancreas of an obese individual who came to autopsy. And what it shows is that the black stained cells are insulin-producing cells. And you see that they are budding like a flower from the ducts in the pancreas. And the individual who's obese has many more of these cells that are being produced from the ducts that are in the pancreas. And that's recapitulating what happens during embryogenesis. That's where the beta cells come from. They come off those pancreatic ducts. And the obese state is one where you need more insulin, and so these cells that produce insulin grow out of the ducts. The problem, though, is that obesity is not a cure, because look what happens with the passage of time. The individual then begins to no longer compensate, and those extra insulin-producing cells die off and refuse to continue to be produced. So it's a transient beta cell growth seen in obese individuals. More importantly is to answer the question of how you can reproduce what happens during those states of obesity, pregnancy, and gastric bypass. Well, it's unfortunate. You would think, okay, what is it that works in those situations? Well, we don't know, to be sure. So the first question of how can we stimulate beta cells to grow is not easily answered. And it's been trial and error. And the trial and error over the past, I can tell you, 30 or 40 years has been to test a number of different growth factors, some of which are elevated in pregnancy, for example, prolactin. And the list goes back some 30 or 40 years. And when some 25 years ago, I contributed to this research when I was at the University of Miami, we used insulin. Insulin itself can make beta cells grow. insulin-like growth factor, growth hormone, prolactin. The problem with those approaches was that they worked fairly well with cells in tissue culture, but their application to patients is problematic. Growth hormone actually resists any beta cells that you want to grow out by blocking insulin action. What's come up only in the last 5 or so years are the two peptides or hormones at the bottom of the list, gastrin and glucagon-like peptide 1. Those are intestinal hormones, gastric and intestinal hormones, that are quite powerful in getting beta cells to grow, at least experimentally. Let me share those data with you. This is a slide of, I don't have my mouse here. Or do I? I'm one of these technoplegic people that can't progress with all these little gadgets. So I'll try to do my best to explain the slide to you. Okay, in the left upper panel, you see some yellow lines that show that mouse that develops type 1 diabetes has become progressively more diabetic. In the right lowermost panel, you see that those yellow lines all go down towards normal blood glucose levels. And the reason that those diabetic mice were protected from hyperglycemia and became normal glycemic was because we treated them with those two hormones I told you about from the intestine, gastrin and glucagon-like peptide 1. Okay? So this is an animal model that develops type 1 diabetes, that mouse does, and it's cured after only 3 weeks of being injected with those two hormones, gastrin and GLP-1. And the cure lasted for some weeks after the treatment was discontinued. This was remarkable, and we published this in 2008. And this shows you on the left panel that there are some brown-stained insulin-producing cells before treatment, but with the passage of time, when you treat with vehicle, you can't find any more insulin-producing cells, and the mouse is severely diabetic. By contrast, on the far right, you see many brown-stained insulin-producing cells that cure the mouse, having been treated with the glucagon-like peptide 1 and gastrin. And on their higher power, what was even more surprising was that the decrease in the brown-stained cells from before treatment to vehicle, which was prevented on the far right by treating the mice with GLP-1 and gastrin, was achieved without the blue cells that surround the islet coming in and killing the new beta cells. We had stopped, apparently, the autoimmune attack delivered by those blue-stained leukocytes. Okay, so we got double the benefit. Not only did we regenerate new beta cells and cure the diabetes, but we did not have to use immunotherapy. Somehow those two gut hormones were able to stop the autoimmune attack from killing the new beta cells. So in summary, what I've said to this point is that the combination of these two natural intestinal and gastric hormones, GLP-1 and gastrin, regenerated new beta cells in the mice, protected them from being destroyed by the autoimmune response. Well, that's all very well, but the question now raised is, are we really going to start injecting our patients with insulin and two other hormones, 3 different kinds of injections, as we had to do with the mice? Or is there a way to increase the body's own production, because I call them natural hormones, of GLP-1 and gastrin coming from the intestines in our patients? Could we increase those levels by blocking their clearance from the body, by using inhibitors of destruction? And the 2 inhibitors that are available, and they're out there in the pharmacy, surprisingly enough, are called DPP-4I, dipeptylpeptidase 4 inhibitor. That's a pill. It keeps your blood levels of GLP-1 elevated. And the other one is a proton pump inhibitor. That's an antacid. You don't even need a prescription to buy it. And that will increase the level of gastrin. Putting these two together in the mice reproduced what we achieved by injecting the two peptides. The yellow column shows you how many more beta cells we had in the mice compared to the white column when they were not treated. And the number of, or rather the numbers of beta cells in yellow is 50% of normal. That's more than enough to cure diabetes in the mice, which was what we observed. And our final experiment was, what about human cells? It's all very well to cure type 1 diabetes in a mouse. So what we did was we got from donor pancreases, okay, the same pancreases that were used to isolate islets for transplantation. Our surgical colleagues provided us islets and other cells from the pancreas of adult humans that were pancreas donors. And you see there are many different kinds of cells in there. Some are beta cells and some are other cells. And what we did was we put these cells into a mouse that has no immune system. It's a living tissue culture vessel. It can accept human cells. And we treated the mice with our two inhibitors. Remember those two pills that cured the diabetic mice? So we asked the question, would those two inhibitors raise blood levels of GLP-1 and gastrin in these mice that are now harboring adult human cells? And on the left, you see day one after implanting the human cells under the kidney capsule of the mouse pancreas, very few brown-stained insulin cells. 18 weeks later, because there's a good blood supply under that mouse kidney capsule, many more brown cells in the middle panel appeared. But best of all was when we had those mice treated with the two inhibitors. Then you get many, many more insulin-producing cells, and the yellow bar shows how many more. This slide shows you where those new insulin-producing cells came from. The left panel shows in green staining insulin-producing human beta cells. Okay? In the middle panel shows the duct cells from the exocrine, non-endocrine exocrine pancreas that I told you about earlier in red. And on the far right, you see superimposed in yellow the fact that insulin-producing cells in green on top of the red ductal cells. Since the staining superimposes and shows up as yellow, it means that the insulin-producing cells generated from the ductal cells in the human pancreas. What I'm saying is that the ductal cells in the human pancreas, consider them as stem cells, if you will, became the source of the new beta cells under the influence of the two inhibitor molecules. And most importantly and finally, what you see here is a graph of blood glucose. Okay? Streptozotocin kills the beta cells in the pancreas of those mice, and they become severely diabetic. By comparison, even though you've treated the mice with streptozotocin, which knocks out their beta cells in the pancreas, in pink, the glucose is controlled now by the human cells that mouse is harboring, and in yellow, beautifully controlled by the human cells that have grown up and proliferated under the influence of the two inhibitors. So that was the penultimate demonstration that if we did a clinical trial using these two inhibitors in people with type 1 diabetes, since they have a pancreas that has stem or precursor cells, perhaps we could turn them on to become beta cells and control glucose. So that's the rationale. Now, little personal story here. I'm a Canadian. Thank goodness for free trade. I'm down here on a visa. And my home, where I did all that work with my team in Canada, was in Edmonton, in the province of Alberta. When T. Denny Sanford made me this offer that it was impossible to refuse, the arrow shows that I came down to Sioux Falls. And at that time, JDRF had supported all of our work in Canada and was prepared to support this clinical trial together with the funds from the Sanford project provided by Sanford Health and Sanford Research. Now, those two kids in North Florida, by the way, had written to us. And you see their little letter. It's so cute. I just love it. Dear mice and scientists. And my staff, of course, answered what the mice eat, low-fat cereal, of course, and where we lived. And on a more serious note, you know, when JDRF talks about discovery, that's the first hill you see there on the left. Okay? And that's all the basic research that I told you about over the last 6 or so years that led to the second hill, which is now development. We're developing what was discovery to determine whether or not it will truly be valid for eventual clinical practice. That would be delivery to patients. So we're in the middle hill now, which is called a clinical trial. A clinical trial means that we're going to, under FDA scrutiny, determine whether or not our 2 inhibitors could improve glucose control, if not fully regenerate and get our type 1 diabetic patients off insulin. And it's called Repair T1D, restoration of pancreatic insulin response in type 1 diabetes. Here's the objective of our trial. The DPP-4 inhibitor is called sitagliptin. It's a pill taken by type 2 diabetics, by the way. And the proton pump inhibitor is called lansoprazole. And these two pills, capsules actually, or placebos, are taken by our patients at the time that they've just been diagnosed. What that curve represents is that by the time people are diagnosed to have type 1 diabetes, that is, are hyperglycemic, okay, they've probably lost 70 to 90% of their beta cell mass. They're down to about 20% of normal, and they need insulin. So our objective with these 2 capsules is to optimally regenerate their beta cells for reasons that I provided in terms of the preclinical work, or at the very least prevent their beta cells that are there from undergoing further damage due to autoimmunity. Because what we do know is that in the white box, the residual beta cell loss continues after diagnosis. And over the first 12 months after diagnosis, there's a good 50% drop-off on the average in what the original beta cell mass was, so that patients definitely have to be taking insulin indefinitely. And our trial looks like this. The first stage, of course, is screening. Now, I'd ask the Food and Drug Administration whether we could do this trial in patients down to age 8. But the Food and Drug Administration said, no, you better start at 11. That was arbitrary. Why not 10? Why not 13? Well, that's FDA. They said 11. Now, why did they not allow us to go to a younger age? Because the DPP-4 inhibitor, sitagliptin, is approved on the marketplace only for adults and only for type 2 diabetes. The proton pump inhibitor was not a problem. So they have to be in that age range, 11 to 45. They have to definitely have type 1 diabetes and not type 2, because we're trying to cure an autoimmune disease, and the way to know it's type 1 is by a blood test that shows that you have certain antibodies circulating. And thirdly, within 6 months of diagnosis, because I showed you the ball game may be over a year or 2 later in terms of residual beta cell mass. We don't know that for sure, but we're going to start with those who have some beta cells, and if it works, we'll open the window and go to those who are out further. The run-in period is critical, and any new type 1 diabetic, we always, of course, have to help with a variety of things, which you, who are family members of type 1 diabetics, are well aware of. Everything from education to nutrition counseling to intensive insulin management to optimize glucose control. So that run-in period between screening for eligibility and assessing them at baseline to start their medications is on the average a month or so. Now, baseline is the all-important point where we do a variety of measurements so that we can determine later on if things have improved since the baseline. And the all-important measurement that you see on top there is the C-peptide response to a mixed meal tolerance test. The patients drink a boost. You can see it in the pharmacy. It's a combination of high protein, high carb drink. And the glucose response to that drink and the insulin response to that drink tells us how well their beta cells are functioning. And of course, blood glucose, HbA1c, and insulin use. They take their medications for 12 months, and the important point to be made is that it's a double-blind, placebo-controlled trial. That means we don't know, some of our nurses are here tonight, they don't know, and the patients don't know if they're getting the active sitagliptin lansoprazole or the matching placebos. However, there is 2 chances out of 3, which we tell the patients, that they're going to get the active meds. And it's completely blinded because the capsules look exactly the same. Every 3 months they come in for testing according to C-peptide response to a mixed meal tolerance test, glucose, A1C, and insulin use. At the end of the 12 months, they're followed up to see if we've had benefit, does it last? Now, we have completed enrollment, I'm pleased to say. And although it's taken a year and a half, the reason being, and you can see in the color-coded bars, being that when we started enrolling here in Sioux Falls in red, by the time we got everything in place and the contract signed, etc., and the team ready, in yellow, Fargo came on board, and they recruited and enrolled patients. And then we got the Minnesota Children's Hospitals and Clinic in St. Paul, in green, to enroll patients, and finally the Rady Children's Hospital in San Diego. So that accelerated the pace of enrollment. And we've got, as you see now, 61 fully enrolled patients in the study. The age distribution of our patients is shown here. The males outnumber the females to a slight extent, and it's known that the incidence, or prevalence, I should say, of type 1 diabetes is a little higher in males than females. It's, of course, highest in the youngest. And you can well appreciate that had we been allowed to do 8, 9, or 10-year-olds, we would have had a far greater rate of enrollment. 75% of our subjects are age 17 and below, and only 25% are age 18 and above, which is fairly typical for the relative incidence. Now, this looks complicated, but I'll try to explain it simply because it's all important. These are the results at baseline of the all-important, how does the glucose rise, and how does the insulin produced by the patient's pancreas respond to that glucose rise? And you can see that our patients sort out into 2 distinct groups. In blue are the individual patients that have a too high a rise in blood glucose on the left. And on the right, it's because, again, in blue, they don't produce a good plasma C-peptide, which is a surrogate for insulin response. So they're really running low on their beta cell mass. Okay? Those individuals who have better glucose control, yellow on the left, is because they have a better insulin response, yellow on the right. So our objective is to see whether those individuals in blue who have a very flat insulin response and a very poor glucose tolerance can regenerate beta cells and become more like the yellow colored individuals that have a better insulin response and a better glucose control. And that we can protect those who have a reasonable insulin response at diagnosis in yellow from crashing down into the blue zone. And that's what I indicated earlier on as the objective. So I'm only the representative of very hardworking people some of whom are in this room, such as Don and Jen, and the team in our clinical research center. And I see Dr. Davis Keppen up there. Am I missing some of our other team members here? Our doctors in pediatric endocrinology, in adult endocrinology, Their physician assistants and diabetes nurse specialists are regulatory people who are responsible for administering and making sure that everything gets done. Because when we have 4 sites, including Sioux Falls, and over 60 patients, and they're seen every 3 months, you can imagine the wealth of data involved. And I know Paul Thompson, who's our statistician, is also there. Am I missing any other members of the team? Because they're really the people who do all this hard work, and I appreciate, believe me, their efforts. Finally, coming back to JDRF, not only did JDRF support my research that I showed you that led to this clinical trial while I was in Canada, but JDRF continues to support this clinical trial, along with Sanford, as I mentioned. And in the past, when we did islet transplantation as a therapy for type 1 diabetes in Edmonton, Canada, I can remember well Jennifer Clare, when she was 11 years old in 1992, at which time JDRF gave us tremendous support for islet transplantation, drew this little cartoon, which continues, I think, to say it all. Any kid, especially with type 1 diabetes, has this huge dinosaur looming over them, and that's what we've got to put out of commission. Thank you.
Okay, so now we can take questions from Dr. Rabinovitch, and we'll start. Can I start in Sioux Falls? If anyone has questions there.
One question that I have, I've been doing government relations for 9 years and following the artificial pancreas. And one thing that seems apparent as the research improves and they find like the new pumps, and then now there'll be the pump with the glucagon, it ends up, the FDA wants a new clinical trial, and it kind of ends up pushing things farther and farther out. So the question I have is, with the regeneration, is what you're doing now, it's kind of variables kind of staying the same, or are you seeing as you go along and you're finding new stuff that these new variables come and the FDA wants a different type of clinical trial? So, because I'm kind of thinking regeneration will actually be able to get to the market quicker than the artificial pancreas, just because FDA doesn't, they have issues with all the different mechanical parts.
Okay, the answer to your question is really simple. It all depends on success. If there's any evidence, even if it's not statistically significant, sorry, Paul, if there's a trend that these agents, okay, lower glucose, lower HbA1c, improve insulin secretion, if it goes in that direction, then in no time at all we are permitted to engage many other centers in doing a phase 3 clinical trial with hundreds of patients, okay? So that is even necessary before FDA is going to approve it as an investigational new drug application, okay? Sorry, it is an investigational new drug. Approve it as a new drug application.
Very exciting work, Dr. Rabinovitch. I understand why you're using patients that are early diagnosed because they have a reasonable residue of beta cells. I guess I'm trying to get at how broadly applicable you think it would be to other diabetics that are further on down the line. They still retain some level of residue beta cells in the population.
Oh. I'm very confident that we can open the window to patients who've been out longer. And I can say that now because I know of another clinical trial using an immunotherapy, not related to this, where there's been some trend of benefit. And what the investigators have now been able to do under a new drug under an investigational new drug application to the FDA, they've been able to open the window out to 2 years. So you're asked first to do it on those more likely to respond. And if you got any trend, you quickly are accepted to go out, yes.
I guess do we understand now that, say, if somebody's been diabetic for 20 years, do they still have some level of beta cell?
Yes. The answer is yes. But as I just pointed out, perhaps regeneration from the non-beta cells will work. You may not even need any beta cells for it to work. At least in mice you didn't, but in people it remains to be determined. But we're not going to abandon the large population of long-standing type 1 diabetics, not at all.
Any other questions in Sioux Falls? It's back to you, Jackie.
Okay. Do we have any questions in the room here? Sure.
Thank you, sir. Are you familiar with the Faustman labs and what is happening at Mass General with, I believe, an anti-TB type of drug that's being looked at for regenerating beta cell function?
I know he is, so he'll be back.
What's cooking in the kitchen?
It's all right.
I'm just curious how often the oral agents are taken. At the same time they take their insulin, they're taking their oral agents as well.
The patients are certainly continuing their insulin. Okay?
Right.
And the capsules are taken for breakfast. Well, that's arbitrary. We've had a couple of patients
right on where they found it easier to take it in the evening because they remembered better, for some reason that escapes me, that they would not fail to take it by taking it in the evening.
But so it's once a day?
Once a day, yes. I'm sorry, once a day.
Thank you.
Dr. Rabinovitch, we did have a question in Minnesota. Someone was wondering if you were aware of Dr. Denise Faustman's work using a drug that was really approved for the treatment of TB, and what it's showing and how it's related or not related to what you're doing.
Yes, I'm very well aware, and I've known Denise for many years. It's very interesting what she's doing. Our group in Edmonton actually discovered it many years ago, and that's why I'm excited to see whether Denise's work will pan out. I know that she's doing early trials right now.
And is it similar in that it's a similar protocol? Not similar protocol because I know that it's a different drug, but it's a delivery of oral medication that's intended to accomplish the same things as your trial, or is it intended to accomplish different things?
No, it's an injection of a form of non-infectious tuberculosis material that she's testing in patients who've had diabetes for a long time. And previous studies have not been effective in reversing diabetes in patients, but maybe some of the new work that Denise has brought to consider in her trials remains to be seen, and we're looking forward to perhaps some benefit there.
Great. Do we have other questions in our room here? How about, did we have any in the conservatory? Okay, so how about additional questions for Dr. Pragnell? Oh, wait, there is one in the conservatory. Hang on.
I'm just curious, if you're treating these patients with anti-resistant drugs, what would stop someone from just going out and getting them and taking them to see if it worked?
I'm sorry, could you please repeat the question?
So someone was wondering, since these are drugs that are available on the market, what would just prevent somebody from going out and taking them and just seeing if it works?
Nothing whatsoever. The problem would be that the particular dose, timing, and knowing by the specific measures we're doing that the beta cells are coming back. So this is typical of a trial. You have to have criteria regarding the drug you're using and the assays that you're using to measure the effects of the drug, and done in enough